It is my utmost pleasure to be here today with our two speakers. I would like to start with the acknowledgement and to briefly introduce the speakers later. So I wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca and the Mississauga of the Credit. Today, this meeting place is still the home of many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. I also am privileged to be one of the co-chairs of the T-CAIREM Center and the Temerty Center for AI Research and Education and Medicine at the University of Toronto. It has been made possible by the generous donation of Temerty family, to whom we are very grateful. T-CAIREM is an interdepartmental center that serves as a focal point for collaboration among healthcare providers, trainees, researchers, computer scientists, engineers, and industry. And our goal is to transform health through AI. For those of you who need this information about the accreditation, this event is CPD accredited. To obtain your credit, please complete the evaluation form that we will email all the attendees following the event. And don't forget to provide your name and email address for the evaluation form. And now, also a quick note for all of you, if you have questions to the speakers, use the Q&A function in the Zoom to submit those questions, and they will be addressed during the discussion portion of the webinar. This event is also being live tweeted, and you can follow it on Temerty Talks. But please go ahead and listen to these wonderful speakers. As I said, it is my utmost pleasure to welcome today our two speakers, Mary-Louise Greer and Moti Freiman. I have met these amazing researchers at the T-CAIREM event in Israel, and they have put forward an incredible project that we are very excited to help to hopefully bring to life. Mary-Louise Greer is a general pediatric radiologist on staff at CKIDS in Toronto since 2011, where she's a co-section head of a body MRI and an associate professor in the Department of Medical Imaging at the University of Toronto. Mary-Louise currently serves on the MRI committee of the Society for Pediatric Radiology, the Pediatric Review Panel for Radiographics, and is secretary of the Pediatric Study Group of the International Society for Magnetic Resonance in Medicine. She teaches, and she enjoys teaching and research, and has published many papers that are highly acclaimed. Her interests include imaging and oncology, inflammatory bowel disease, application of advanced techniques in MRI, and the evolving role of AI/ML and the promotion of greater access to pediatric MRI through non-sedation strategies and outreach activities. And I will briefly mention Moti's also the background before I pass on the baton to Mary-Louise to speak first. Dr. Moti Freiman is an assistant professor and the director of the Technion Institute of Technology Computational MRI Lab at the Faculty of Biomedical Engineering of Technion in Haifa, Israel. He's also an academic director of Technion Human MRI Research Center. Previously, Dr. Freiman held various positions in academia and industry, and he's a co-author of many acclaimed papers as well. He's serving on a program committee member at MICAI and IEEE conferences. He's completed his postdoctoral fellowship at Harvard Medical School in 2012 and holds PhD in Master's in Computer Science from the Hebrew University of Jerusalem. And without further ado, I will pass the baton to Mary-Louise, and looking forward to your talk.
Thanks very much, Anna and Zoriana and everyone on the T-CAIREM and Technion team. It's a great pleasure for Moti and I to be here today and speak with you. I'm here in Toronto. Moti is speaking to us from Haifa. So we hope we don't have any technical glitches. So to begin, we have, sorry if I can just advance it there. There we go. We have no conflicts, but we do have the following disclosures. I'm going to shrink my screen here. I can hopefully, there we go, see the screen better. Hopefully you can too. So the majority of this is going to be an overview of the program that we were fortunate to be part of, and the second part being the scientific component. The learning objectives from the scientific component of our presentation will be hopefully at the end you'll be able to understand the role of quantitative MRI in assessing inflammation, to appreciate the potential of AI tools for quantitative MR analysis or inflammation assessment, and to identify specific quantitative MRI biomarkers in the setting of inflammation assessment in Crohn's disease in particular. So I'll be presenting first on an overview of the inaugural T-CAIREM Technion collaboration Catalyst workshop that was held in Israel in early May of 2023. It's a bit detailed, I'm certain. I'm sorry for the second part, but this will be specifically about our combined projects of the different researchers and the one for which we are fortunate to be grant recipients from T-CAIREM Technion, and that is to discuss the potential role of AI-based quantitative MRI to identify clinically relevant microstructural changes for clinical application in pediatric Crohn's disease and potentially trigeminal neuralgia. So to begin, I'm going to share with you over the next few minutes all about the Catalyst workshop. And what were the goals of this? Well, the first thing was to expand scholarly ties between the two institutions, to facilitate academic cooperation amongst faculty members between both institutions. And I'd like to highlight here that it's also achieved an unspecified goal, which was that came out of this, was that within our respective institutions, both Moti and I have been very appreciative of the chance to get to know researchers within our own institutions that we previously did not have any opportunity for engagement. So it was both within and between the two institutions was a great opportunity for collaboration, getting to know each other. To really take advantage of that, to draw on the expertise of the faculty members to increase education opportunities at both institutions, and us being here as an example of an outcome, to enrich each other's educational and research environments, and particularly to promote international and intercultural understanding. And I think that was achieved to a great extent. I really have great pleasure sharing with you some of the photo journey, and it's not often I get to share my holiday snaps with such a great audience. So the people whose brains were brought this to fruition were Mohammed Mamdani, who amongst his many roles is director of T-CAIREM here at U of T, and Danny Aiton and Shai Shanor at T-CAIREM. The support crew, you've already met Anna, and also Amjay Masui, and you've met Soriana, and on their side Yolanda. Yolanda, I didn't have a photograph of you, and then Tal Shalem supporting the Technion team. So what happened? Well, a call went out in February with a closing date in March to participate in this great opportunity. We were given the instructions about what to do and what the outcomes were likely to be. And the thing that was particularly relevant was which faculty were eligible within the Faculty of Medicine. And so there were clinical areas of focus, and you can see here, I'm a pediatric radiologist. So this was really speaking to me as someone in pediatrics and medical imaging. And on Moti's side, he's has vast experience in looking at medical imaging data and core learning and deep learning. And he and I have had opportunity to collaborate through other multi-center projects involving IBD, inflammatory bowel disease. So we felt that this was something that we were very excited to potentially have a chance to participate in. And within 3 weeks, we got the great news that we were able to participate in this great collaboration. And the other exciting aspect, which showed great vision, I think, was that we were able to invite our trainees to participate. So I brought one of my trainees from Toronto, and Moti had one of his from Haifa. We had to provide relevant information so that we would all get to know each other in advance as well as at the time. And I like to call this whole experience the Ein Gedi Big Brother, who was thrown into a fairly remote environment and spent a concentrated period of time both on the science and sharing meals, and it was an amazing experience. These are the team from Technion who got to be part of this great program, and you can see people, trainees and faculty from different aspects of medical and science from Haifa and Technion. And this is the group who came from Toronto, again, from many areas of medicine, science-based, clinical-based faculty and students. So in the end, the group that made the trek from Toronto to Israel, 12 faculty with our T-CAIREM leads and adjudicators, 7 trainees and 1 administrator. Our first day saw us arrive late on the Saturday, and this was the group that was there in quite a wonderful environment. The next day, in order to allow us to get over the jet lag and acclimatise, we were treated to an amazing experience of going from Ein Gedi to Masada National Park and then back to Jerusalem. Everything we needed to know was provided, and here are just a few snaps of what was a really incredible experience to this very historic location on the hills overlooking the Dead Sea. And here's I am Zoriana, and my trainee here is, and that's Farah and all of the group here. And then from there we went on to Jerusalem, and you can see here at in the middle of the old city, and the 2 mics. There's Mike Frolick and Mike Rodno. And so that was a day for those of us from the Faculty of Medicine who hadn't really known each other before to have a chance to bond. While we were making that journey around the shores of the Dead Sea and back to Ein Gedi, our colleagues and soon to become friends were making the same journey from Haifa through to Ein Gedi, and work really got underway on the 8th. So it took us a little while to all get together and then and become acquainted. And that first day was really about that. We had an opportunity to share short presentations to outline who we were, our areas of research, and things that we thought were potential opportunities for collaboration. So the faculty did the presentations, and through the course of breaks and dinner and the like, we all got to know a little bit more about each other and work out who we might have commonalities with, that we might be able to share experiences and work on projects together. So that night and the next day was really when we started to form teams. So here's an example of one of those presentations, though, on that first day on the Monday. So the things before we formed teams was the sharing with each other, looking at collaboration opportunities. And I think it's important to highlight that part of this was also being mindful of aspects of responsible AI. It's a beautiful environment that we got to do all this in. And there was lots of opportunity for social networking. So this is day 2 when our teams had been formed and we started to work on projects. And we had our goal was to really come up with a project that involved members from both of the institutions and multidisciplinary teams, and come up with a 2-page summary of what we wanted to do. And so here's an example of our team. You can see our students, Tim and Farah, and some of the other PIs. So Moti here and Moji here is a neurosurgeon. And we've got Noam, whose area is genetics, and Rafet, whose area of expertise is psychology and computer programming. And this is what we had to come up with our précis for the 2 pages. But we were really, the whole part of it was really to improve engagement. And I think that the opportunities, not only to share our scientific enthusiasm and ideas, but really to get to know each other as individuals was quite wonderful. And it was not something I've experienced myself before. So at a certain time of the day, there was that, no, that's it, stop. We really want you to get out and just get to know each other outside of this environment of just the research side. I can see contemplating in this environment. It was Moji here, going to the Dead Sea. So our Haifa colleagues threw us all in cars and took us down to an area that we could swim, float, make sure for Tim got an early casualty, rolling around, getting salt in his eyes. You can see these are on the, getting in the shade here. And this was the team. So it was really, really quite an interesting and spectacular occasion to really get to know each other. Later in the day, after we had shared another meal together, there was a surprise that was held. That was a pool party and a magician. And Moji decided this is her favourite experience of the whole thing, was having to write a name, put it in her pocket, and have the magician tell her what that name was. And some of the other members of the team who had fun and games. And then the work concluded on day 3, where we then, again in this great environment, our 6 different teams that had formed over that period of time had to present our project and work out, which ones were likely to go forward, and these are some examples of the presentation. So Alan was presenting with his team. You can see we have here's Moti presenting our project. We have William Tran here presenting, and some people knew how to work the room really well. There's Will working his stuff. And then you can see then Michael presenting on cardiac, and he's also not only a cardiologist but an artist as well. And some people are really great storytellers. So here's Mike Riley with his presenting his team's work, and Melanie, saying it's all about the data. What about the data? And so we had a fun in the midst of all of the seriousness, and poor Shai and Mohammed then had to know what on earth do they do with all of this. So they called in some help with the adjudicators, and you can see Anna here and MJ and I think Danny here, having to work out the pros and cons and the likely feasibility of what we'd pulled together in a very short period of time. And so they had to decide who the grant was going to go to. And the fact that Moti and I are here presenting with you suggests that, yes, we were fortunate to be the grant recipients, much to our surprise. And it always helps to have the trainees who know how to do selfies present that. But the great thing about it is not only the opportunity to really bring together people from very different disciplines. So again, genetics, psychology and computer programming, neurosurgery, our trainees here, medical engineering and pediatric radiology. But together we were able to come up with a project that we're going to share with you a little bit more. But the other thing that's come out of this is not only was our Ein Gedi crew successful, but there's been a great push for those other 5 projects to also be able to get some grant funding to move forward. And just before we started this presentation, Zoriana shared with me that William, who is working the room so well, his collaboration team from this workshop has been successful in getting a grant for data sets analysis. So at the end of the day, after we had some more opportunities to bond at the shores of the Dead Sea, not so easy to swim in that area, but quite stunning scenery, and really was again a great chance for us all to kind of share this experience together and support each other through. And I think that this really sums up to me that proposal that was posted in February about bringing together groups of researchers from T-CAIREM and Technion to really build this great collaboration was very successful. If nothing else had come out of it, just what we achieved at the time was flagged on the Jerusalem Post, one of the main online news services there, the day that we were all heading off back to our respective homes, as being the AI solutions in the field of medicine, and the article that went with it. So it was incredibly special opportunity and a real privilege to be part of this. So at this point, I am going to hand over to my co-investigator and great friend, Moti Freiman, and he's going to share with you more of the science that's has come out of this, some of the stuff that's been a lead-in for our respective areas of interest that we brought into this project. So Moti, I'm happy to hand over.
Thank you. Thank you, Mary-Louise, and thank you everyone for joining this session, and obviously for inviting me to speak. It's indeed, it was a great pleasure and a great experience. So our team was composed from 5 researchers, 2 from the T-CAIREM, Mary-Louise, who just presented, and also Moti who is a brain neurosurgeon and scientist. From the Technion side, we have myself working on AI for medical imaging and specifically for MRI, Rakefet, who is working on metacognition, and Noam, who is a biologist interested in epigenetics. So we put slides. We continue actually the collaboration. So we maintain some visits in Toronto. I visited with my team during the past SMOM, and Rakefet was in Toronto during August. So basically, we came from different disciplines, and one of the challenges that we were faced was how to create a story from our unique interests. So Dr. Moti is working on pain, and specifically on trigeminal neuralgia, and interested in pain. And basically, in the past, pain was been defined as a subjective condition. But that's imposed significant limitation on diagnosis and prediction. But with data science analysis of tissue microstructure, we may understand pain better and can may have an opportunity to change this. So what we would look like from her perspective is how data science and analysis of MRI images using AI technologies can be useful in better understanding pain and predicting outcomes of some treatments. On the other hand, Rakefet was interested in metacognition. So medical diagnosis is a cognitive challenge, and we think about how radiologists making diagnosis when they are looking on images, but we can think how AI tools can improve certainty or reduce uncertainty when making decisions, reduce the level of consultation with peers, asking for additional tests. So all these kind of things can be considered metacognition. And the question is how, from her perspective, is how integrating AI tools in clinical diagnosis can improve or benefit benefit the decision process in terms of diagnosis quality, saving time, journals development, etc. Noam was interested in how molecular epigenetic information regulates genome function. And that's also can be interesting when you look on diseases and also on pain as a phenomena. So disease can occur both at genetic and epigenetic levels. Obviously, the implications are at the macro level, more macroscopic level. And can we analyze this data together so we can measure and analyze high throughput genetic and epigenetic data, looking on the physical 3D structure of the genome, and we can develop computational tools to understand the mechanistic models influence these changes in the genome. Mary-Louise, this is your work, so I'll let you to present it.
Thanks very much, Moti. So the area of interest for me is inflammatory bowel disease in children. And one of the things that I've been working on with my colleagues, also based in Israel and at CPD in Toronto, is developing a scoring system based on imaging features that we discern on MR andrography or MRI of the bowel. And you can see examples here of the items that we have found to be helpful in working out both the presence or severity of active inflammation. And that score has been validated in the last year and published, and it's called PICME, and that looks at wall thickness, ulceration, restricted diffusion in the bowel wall, changes in the mesentery and the vasculature. And so we've now taken that to apply to other aspects of clinical dilemma, and working with my my student Paradin, who's now back in the UK, we were trying to discern different features of that are seen in inflammatory bowel disease that can be very difficult to separate out sometimes from mimickers such as lymphoid nodular hyperplasia. So the lymph nodes are enlarged, and it can be, whilst it's quite straightforward to work out what's normal bowel versus a more severe end of the spectrum in inflammatory bowel disease, it can be very challenging when you have mild inflammatory bowel disease versus lymphoid nodular hyperplasia, which is frequently a normal variant, but may not necessarily require treatment as you would expect for Crohn's disease. And so we have applied the PICME as another way of trying to better discern what may be inflammatory bowel disease versus lymphoid nodular hyperplasia to show that that's of added value. What we'd now like to do is then take some of that, those ways of analyzing this terminal ileum, a small part of the bowel which is often affected with Crohn's disease, and starting to apply some of the tools that we are talking about in terms of AI, improving reliability and confidence, and separating out those entities. So I'll hand over back to you. Thanks, Moti.
So thank you. When I started to work with Mary-Louise, so we came with a very basic thought that designing or developing indices to assess the severity of Crohn's disease, radiologists tend to use linear combinations, only a weighted sum of this item. So with Itai, a student of mine, we developed a machine learning model basically taking the radiologist assessment, the different items like wall thickening, narrowing, enhancement, etc., and rather than combining them naively as a weighted sum, we can develop nonlinear machine learning models for that. And actually, in a paper that we published a year ago, we show that by using machine learning, nonlinear machine learning models to develop more complex indices, we actually better predict surgical or invasive assessment outcome, like here you can see with gastroscopy, ileocolonoscopy, sorry, you can use MRI combined with machine learning models to understand the connection between the radiologist assessment and the invasive assessment. So that was a first part of the work. And now I would like to change to a more advanced MRI-based approach to assessing inflammation. So beforehand, we saw a lot of images, and radiologists are looking on images all day, but these are qualitative images reflecting different contrast between tissues. So when you see something bright or something dark, what that means, what is the biological properties that we are actually measuring? This field called quantitative MRI. And in quantitative MRI, we usually take multiple images, a series of images, while changing some of the acquisition parameters. So in this example, it's diffusion-weighted MRI, where the parameters that we are changing during acquisition called B-value. I won't go here into the details of the imaging physics, but you can see from the image that the signal at a specific voxel is decaying with some kind of exponential shape. And the interesting properties that we can measure when we fit a model to this decay is actually a biological property of the tissue, and we can actually quantify it. So in a specific model that was proposed by Dennis Lebian called the intravoxel incoherent motion, and again, I won't go too much into details, we have here a B exponential model that characterize different properties of the tissue. So one is the either restricted or unrestricted diffusion, the pure diffusion of water molecule within the extracellular space, and that considered related or indirect measure of tissue cellularity, which is an important property of the tissue. We can look also of F, and F, the parameter, measures the percentage of the microvascularity within the tissue. And you can look also on the third parameter, which is D star, which presents the blood flow within the microcapillaries. And these three parameters can provide you a quantitative insight to tissue properties associated with multiple clinical entities that we're interested in. So in the context of inflammation, people tend to measure an overall slope. This is called an ADC. Radiologists are usually familiar with this concept, but for sure MRI radiologists. But this measure encapsulates different properties of the tissue. And unfortunately, hyperemia on one hand, and increased number of inflammatory cells, dilated lymphatic channels, etc., are contradicting phenomena that influence the signal in the opposite way. So if you take the average, the ADC value, you basically encapsulate and lose the information you're actually interested in. And your goal, our goal, is to tease out the fast diffusion component, the F, the fraction of it, and the slow diffusion component, which is associated with the number, for example, of inflammatory cells. So the question is how to do that. This is a big numerical question, and people wrote algorithms to solve this problem, how to fit this multi-exponential model to the observed data. We don't have that much data. It's always noisy. And during my postdoc, I wrote one algorithm for that called the fusion booster proof. It's a patented algorithm published in Medical Image Analysis. And with the development of AI models, this is a classical tool, but with the development of AI models, we moved to using neural networks to predict this, to basically to fit this model, this kind of models to the data. So one contribution was by Noam Korengut, a student of mine at Mikai 2022, but multiple other groups has developed multiple algorithms and numerical tools is a classical on AI to solve this problem. The interesting thing is that it's really influenced the diagnosis of Crohn's disease. So in a study we did in the past, we look whether the major factor in assessing Crohn's disease is the D parameter, which is the cellularity of the tissue, or the F parameter, which reflect hyperemia. And I won't go on the details of the experiment. Like, we took a group, and what we found, and this is the important thing, it was published in JMRI 2012, so quite a while ago, but we were first to show that actually what people saw to be reflected by the ADC is actually related to the F parameter, means in inflammation, we can measure with MRI the hyperemia of the tissue, which is an important factor. And actually, this is a factor that distinguishes between groups. And based on this finding, which actually was replicated afterwards by several other independent groups, notably a group from NYU, we got funding from the NIH to see whether we can differentiate between active and chronic inflammation, which is a major question in treating Crohn's disease. And what we show there is that basically, yes, if you look on the fast diffusion fraction, the F parameter reflecting a hyperemia, then you can separate better between active and chronic inflammation. And this results shows that the F parameter is very reflective. It's a very good biomarker to identify different groups of fibrosis score and also acute inflammation score. So that was an interesting finding, and that reflects how we can use quantitative MRI biomarkers in this context, diffusion-weighted MRI biomarkers, to assess specific entities related to inflammation in Crohn's disease. On another aspect of the same idea is to look on T2 distribution. So T2 is other MRI technique where we measure a property of the tissue called T2. It's a more complex property. Again, I wouldn't go in details to the physics of this phenomena, but it's a very important physical phenomena. And people tend to use a single number, a T2 number, to represent a pixel in the tissue, and then obviously you encapsulate the real information. But you would like to, what you actually do, you would like to, is to look on the distribution of T2 values within the voxel. Again, voxel is quite large, so 1 millimeter by 1 millimeter by, let's say, 1 millimeter, and we are speaking about properties of molecules. So obviously there is a distribution, and when you look on for a single number, you kind of are taking an average, which is represented here by the red line. But obviously the red line is kind of hiding the two components of the mixture of Gaussian that is actually representing the different phenomena within the tissue. So if you look on, this is an MRI of a mice, and if you do what conventional T2 mapping, so you can say, okay, this is the bone marrow, this pixel is part of the bone marrow. But if you will look on the distribution, then you can say 60% of it is the red bone marrow, and 25% is blood, and 15% is the yellow bone marrow. So we actually can provide, by doing this complex analysis, microscopic information related to the different components within the tissue. And this is really important in assessing inflammation, as we will see in a few minutes. So we wanted to do some experiment on that, and we took mice, basically 15 animals, 5 at each group. So we have control group, 5 animals with acute inflammation, and 5 with chronic inflammation. And the question was whether we can develop quantitative T2-based MRI biomarkers to distinguish between these 3 groups. And obviously there is an experimental aspect of that, and that was not done by my lab. That was done in collaboration with Professor Catherine Van Dorne, which is a preclinical imaging scientist in our faculty. And what I was interested in, the mathematical aspect of it. So how you do extract this T2 distribution from the MRI images that you acquire. And again, I wouldn't go that much to the equation. It's a very challenging mathematical problem, and there's various ways to do that. And people in the past tried to do parametric solution using non-parametric solution. with nonlinear least squares problems, solvers, and variable projection approach. There's a variety of classical approaches to that. None of them really works. In the past few years, people again started to use AI models to predict this T2 distribution. So in a paper published in Medical Image Analysis in 2021, a group published an algorithm called MIML to predict T2 distribution from the data. We extended their approach in some way. Again, I won't go to the details. We called our specific model P2T2 model, so physically primed T2 model. The main idea was to integrate the acquisition parameter into the neural network. So usually, people think about neural networks as a data in, prediction out. But our data has some physical meaning. So we need to encode in some way the physical setup of the acquisition within the network. This is not a trivial task, and this is what our research did in that paper. So we published it in Computerized Medical Imaging and Graphics, a work that was done by Hadass Ben-Atiyah. And we were able to recover T2 distribution using our approach. So we asked ourselves, what is the implication of using our approach compared to conventional AI approaches in recovering T2 distributions? So we did an experiment. We look on conventional AI, our P2T2 model, and some variation of it that include statistical bootstrapping. And what we found that there was changes in the distribution, in one mode of the distribution. So usually this is a B model Gaussian distribution. And now when you look on one of the modes, you see a trend of decreasing of the value for the between the controls, the LPS group, and the STZ. So, but when you look on T2 scalar mapping, so just averaging basically all the distribution, you won't notice this trend, which means first, it's important to look on T2 distribution when you would like to assess inflammation. And more importantly, when you use different methods, like the MIML approach, the P2T2, and the P2T2 enhanced by bootstrapping, you get better effect size larger by order of magnitude compared to conventional AI approach when you would like to discern between the controls and the other groups. Now, we didn't reach this significant level in post-hoc analysis in distinguishing between the LPS and the STZ, so between active and chronic inflammation. But as you can see, we are not that far away. So we had just 5 samples at each class. What that means that if we will increase our sample size to about 10, then we can pass the significance level. And this reflects the importance of not just using, first you need to use T2 distribution. So you have your imaging information, provide you have the most precise information that your imaging modality can give you. And the second, it's very important to have the right algorithm, or the best algorithm, let's say, to extract the quantitative information from your data. And in that sense, our P2T2 bootstrap did the work way better compared to the baseline AI approach called MIML, and even better that our first attempt, which was called P2T2. So on the technical side, encoding the physical parameters of the acquisition when do deep learning on imaging data has a lot of benefits compared to use, I would call it, naive AI approach when you just fit the images, ignoring the physical context or the acquisition parameters of these images. So these are the conclusions that we have. And that brought us basically, so this is what I used to do. And that brought us together in Ein Gedi to put an application or a proposal called AI-assisted MRI fusion model for characterizing of inflammation. And basically, inflammation is a process that influences clinical outcome regardless the disease. So you can find multiple disease entities, but the origin or both diseases stems from the same biological process, inflammation. And right now, we don't have in vivo tools to characterize inflammatory processes. We have imaging tools to characterize structural changes, but inflammation is a process. So we need to develop it. And the idea is that imaging, as we say it, epigenetics and pain are independent or dependent expressions of inflammatory processes within the tissue microstructures. So the question is whether we can design an AI tool to extract the microstructure inflammation that is the cause to all these implications, pain, etc. And so we started with the idea to first have an AI model to segment the disease region. We would like to analyze a specific region of the images. For that, it's a conventional AI approach, so we will need a manual segmentation to train the model. Then we would like to extract imaging information from this region and also use epigenetic data come from the same patient, same region, to predict the site and the severity of the inflammation. And the third term was to ask ourselves whether this tool actually helps radiologists, not replacing them, but actually how it helps them to use these AI tools as a supporting tool in making decision or reporting the severity of disease as part of the clinical assessment. So, so in terms of innovation, I think there is no AI model that to characterize inflammation. Again, as a source cause or the root cause for multiple diseases. That's a main point. And incorporating of epigenetic material is also an interesting aspect and innovative. So people, what we call now in the AI community, multimodality images. We are not speaking just on language, just on images. Here we try to combine the language of the genome or the epigenetics language and images together to predict outcome, and assessing the clinical impact of such a model, not just by terms of accuracy, but also by terms of how it influences the radiologist or clinician confidence in making a decision. Obviously, the impact will be to improve our ability to non-invasive characterize inflammation in MRI. It will be applicable to a variety of diseases, regardless the anatomical site and patient population. Obviously, because the interdisciplinary nature of the team, so neurosurgeon, radiologist, metacognition, medical imaging, AI, and epigenetics is quite unique and heterogeneous group of researchers, which make it way more interesting, I think. And in terms of methods, we would like to collect data. We are now in the process of establishing the data sharing agreements. And we would like to train our models. Once we will have the data, we will develop the right AI models for that. And then we would like to deploy the models and then perform the metacognition analysis to see how radiologists and clinicians actually benefits from these tools that we are developing. So this is a timeline of that was set up in the proposal. I'm not sure what is zero here, but let's say that it's already started. We are in the process of making the data sharing agreement. In between, we are developing AI models, and we will need to tune them to this specific task, and then we will go forward. On the long term, one interesting aspect of that, when we are looking on two separate diseases, right, Crohn's disease, which is an inflammatory bowel disease, and trigeminal neuralgia, which is a brain disease, we can find an interesting connection between the gut and the brain. So understanding how gut-brain axis regulating inflammatory responses can be an interesting question that would be feasible to explore while we have this unique database and tools. And obviously, we envisage future application both in bowel fibrosis, a very important aspect of inflammatory bowel diseases, and neuroinflammation. Obviously, AI advancement in terms of developing physically primed deep neural networks models to assess imaging data, and hopefully apply for additional external grants such as the CIHR call. This is an eye picture I took, I guess, right? The first day, the last day morning before the talk. I didn't do it today, but it was a nice hike. I go all the way in the mountain, and this is sunshine. It's a very nice place. And again, before taking a question, I would like to thank all the T-CAIREM team for arranging this amazing workshop and for inviting us to present in this lecture series. Thank you very much.
Can have the virtual applause.
Yeah.
Thank you very much for your talks, and thank you very much for highlighting the event. So for the participants, you can put in your questions in a Q&A portion of the webinar. And while you are doing that, I think one of the questions came ahead of the webinar, and the question was how to follow in your footsteps, how to achieve what you have achieved. I think it would be great if both of you would comment, since you had different paths.
So I think it would be good to, there are different aspects for that. So one thing I would like to acknowledge that me and Mary-Louise met in 2012. I would say by chance, right? Somehow, and so working with good people is an important aspect, and not with just working with people in your discipline. My education is computer science. I'm not a radiologist at all, and definitely I'm not a pediatric body radiologist. And it's important to notice to the opportunities that come from other disciplines. So combining AI and MRI imaging and body imaging and inflammatory bowel disease can happen just because both Mary-Louise and me were open-minded to collaborate with people from other disciplines. And I think all the mission of T-CAIREM is kind of encouraging such a collaboration. So just use T-CAIREM resources. I think it's an outstanding opportunity. And the second, yeah, Mary-Louise.
No, go ahead, Moti. Go ahead.
So work hard, and I think understanding the basic mechanism and not just apply AI as a tool to a clinical or medical problem. So understand the sources of the medical problem on one hand, and understand the mechanisms of AI on the other hand. In other words, do MD PhD, I don't know, is a good way to go. This is the only way you can solve real problems. Otherwise, you you solve a problem, but there is no problem that you are actually solving. So you develop a good algorithm, but it's useless. Or you have you don't answer, you ask a question that is not feasible to answer by AI. AI cannot do everything for you. It can do a lot, but not everything. So this is, I think, important aspects.
And I'd like to echo Moti's words that I think having an openness and curiosity, and it really was, one of those things that MRI and working with our collaborators on inflammatory bowel disease project, we heard mention of Image Kids and developing the index for PICME. That really gave us opportunity to meet each other. But really, it was when we had opportunities at meetings to just go and chat, and one or other of us had a post or a presentation, and being there to hear what the other person had to say and support each other and really just open up opportunities and ideas, I think is really important. I come from a very different background. I'm I'm a clinician, I'm a pediatric radiologist, I don't have a PhD, but I have been really fortunate to learn from the people around me. And I think that the thing that I mentioned at the beginning is that this collaboration has opened up opportunities for us to get to know each other within our own institutions. I've showed some hopefully fun photos of the different people who participated in this, and our ongoing collaborations and friendships even outside of our Ein Gedi research group that has come from that. And Moti said the same thing, that there are people within his institution that he didn't really get to know before this happened. And I think it's building those bridges, sort of really bursting us out of silos, and I have always felt throughout my professional career that I'm a pediatric radiologist who does body imaging and MRI. So I deal with the kind of like the thin edge of the wedge in terms of, body parts who move on patients who move. Things always go from, adults to children and neuroimaging to body. So I have to be open to different people who can help me problem solve with my patients and the clinical questions. And as Moti mentioned at the end there, like, for me, the things that prompt my research questions are the things that come up time and again in my clinical practice and when I'm sitting down with my clinical colleagues. And any projects I do, I really like to incorporate the clinical members of the team outside of radiology to let me know what, we're on the right track. And I think that's where Moti and I have really found a commonality and really benefited from our collaboration, is that we each share with each other our experiences and we kind of bounce ideas off each other. And I see all of the great advances that have come out of the last 3 years that have been leapfrogged ahead with things like this Zoom meeting itself. I think let's not lose that. The fact that we can be sitting in different parts of the world today and working together on a project. I can sit there looking at images with his team and say, okay, this is the bit of the bow that we're interested in. So, make the time and take the opportunities, and don't ever be afraid of asking a silly question. That's how I've learned. And great friends have come out of this.
Thank you so much. This is very insightful. We have 2 questions, but only 1 minute. So I'm going to ask the questions. I don't know if you can see them yourselves, but the first question is, do you plan to integrate DTI into your algorithm?
So that's a short answer. It's depend on data availability. We would like to, at least for the trigeminal neuralgia, but not sure that we will have the data. Definitely in a prospective acquisition, I would be very thankful if we will be able to collect DTI data.
Okay.
We have some work on AI with DTI. Yeah. That's to be
And
published.
the second question, can you comment on merging of the new data with the old data as you collect more data and perhaps expand or fine-tune the data and labels? How do you architect and think about iterating the model?
Right. So right now we are looking only on retrospective data. We would like to apply for further grants that will fund prospective collection of data, and then we will be able to collect more advanced data. Then we will, it's a good question how to integrate both old data with new data. There are methods for dealing with missing data, which we may use, but again, it's a challenging question. I'm not sure whether we'll be able to merge the old and new properly acquired data together. We might need to develop independent model for each.
Thank you so much. This has been a great talk, and really enjoyed and really looking forward to hearing more about the project as it develops. Thanks to everybody, the audience, for listening, and follow T-CAIREM. Thank you.